I think we still have an interesting road ahead of us. There's still a lot of unsolved issues. Number one, I think we need to understand how T cells actually work. Once we know how T cells work, why their T cell receptor is so sensitive, then we can translate the lessons out of the T cell world into the CAR T cell world. As long as we haven't reached that state yet, we might just imitate the T cell and change the frame of the CAR and, let's say, turn a T cell receptor into a CAR using the same architectural framework of the T cell receptor, and then see what happens. Now the next thing is affinity. Now CAR T cells have a CAR, and the CAR binds with high affinity to its antigen. The T cell receptor, in contrast, binds with very low affinity, which is still puzzling how this still works. How can a T cell recognize so few antigens based on a fairly lousy interaction? But what the T cell receptor does, if there's just one or two antigens around, the antigen will bind to the first T cell receptor, then move on, bind to the second T cell receptor, and so subsequently activate one T cell receptor after the other. This route is not possible for the CAR. Once the CAR is bound to its antigen, it can't release the antigen. So if you have a limiting number of antigens, you can only activate as many CARs as you have antigens. And that's just not enough to get a robust signaling reaction going. So lowering the antigen or the requirement for low affinity will be important, in my belief, to make CAR T cells more sensitive. In order to be able to do that, we have to most likely move away from current formats of CARs and kind of walk towards frameworks as the T cell receptor is. So turn a T cell receptor into a CAR using basically the same mechanisms, the same underlying mechanisms, and then we can lower the affinity for detection, and then we have this amplification step that happens because of the low affinity interaction between the antigen receptor and the antigen.